Hello everyone, very good afternoon to all of you. Today we are going to see about most excitable video about green fluorescent protein. So the name itself suggests that the protein which emits green light upon receiving the incident light. Let us get into the video detailedly. Our Mother Earth has so much beautiful organism in it. One such organism you are witnessing now is jellyfish. Yes, it gave two important things to a scientific community, though it has a wide pharmacological applications. One is hypersensitivity, we came to know because of this jellyfish. The another thing is green fluorescent protein. It is very much useful in a molecular biology. Fluorescent protein, shortly written it as GFP. It is an autofluorescent protein. It does not require any input. It fluoresces through a biochemical reaction. Little things will make a big impact. Absolutely, it is a very little protein, about 27 kilodalton in molecular weight, having 238 amino acids in its structure, and the genetic makeup of this gene or a protein is about 730 base pair. Our scientist has extracted this protein from a beautiful jellyfish or a bioluminescent jellyfish. Aequorea Victoria, it is entirely a transparent colorless jellyfish, also called as a crystal jelly. It is a bioluminescent hydrozoan jellyfish found near the west coast of North America. It consists of two proteins. One is called as aquarin, a photoprotein, and a green fluorescent protein. Both these are involved in bioluminescence. The scientific classification of Aequorea Victoria is given here. It comes under the domain Eukarya, kingdom Animalia, phylum Cnidaria, class Hydrozoa, order Leptothecata, family Aquaridae. Terminologies which are often used in this video are luminescence, bioluminescence, fluorescence. We added few more things here. The luminescence means the emission of light is not caused by high temperature. The high temperature is the key here. It is also called as a cold light. Chemiluminescence, the chemical reaction which causes the emission of light, that is we call it as a chemiluminescence. Bioluminescence, an enzymatic reaction which is involved in the biological system which emits the light, that we call it as a bioluminescence. So luminescence has two types. Fluorescence, any substance which absorbs the lower wavelength of light and emits the higher wavelength of light, that we call it as a fluorescence. Phosphorescence, it is very similar, but the difference between the phosphorescence and the fluorescence is the incident light. Fluorescence stops upon the stoppage of incident light. But the phosphorescence, in the phosphorescence, even the incident light has stopped, it will fluoresce it will phosphorus for some times. That is what the difference is. In this slide, I gave about the spectrum of light. Anything between the 400 to 700 nanometer, we call it as a visible spectrum of light. Lesser than that, we call it as a ultraviolet radiation. We have a distinguished A, B, C. Then above 700, we call it as infrared radiations. So in this slide, I gave about detail about the spectrum, but in the last part, which is very much important, the shorter wavelength of light has a higher energy and the longer wavelength of light has a lower energy. This, it needs to be understood. After knowing the spectrum, I gave the example of bioluminescence, which takes place in the fireflies because it found earlier. So luciferin is a protein which is present in insects. It respires. The respiration takes place in the abdominal of the insect. The respiration means binding with oxygen and liberating the CO2. So the luciferin binds with oxygen with the investment of ATP, which is catalyzed by the enzyme called as luciferase. So this enzyme catalyzes this reaction. The luciferin goes to the excited state and causes the liberation of CO2. So termed it as oxyluciferin. Then after losing all the electrons, it backs to the ground state. While backing to the ground state, it emits the light. That is, we call it as a bioluminescence, which takes place in the fireflies. Coming to the core part of our video, that is structure of a GFP, because it is a bioluminescent protein. Right, it consists of 11 beta strands which makes up the barrel-like a structure. Alpha helix run through the center of the barrel. So the chromophore is located inside the beta barrel structure. The chromophore is termed as p-hydroxybenzylidene imidazolinone, formed of cyclization and oxidation of 3 amino acids, which is serine or threonine, tyrosine, as well as glycine. 65, 66, 67. These three position is very much important because it causes cyclization and oxidation. The chromophore is responsible for this color formation. This 3 amino acid is making up the color. Here also we come across two terminologies. One is chromophore, another one is called as a fluorophore. A chromophore is a substance or a molecule which is responsible for the absorption of ultraviolet radiations. A fluorophore is inevitably a chromophore. It re-emits the observed light in a longer wavelength. That we call it as a fluorescence. I gave the example here. The substance which observes the lower wavelength and emit the higher wavelength of example is visible fluorescence and the infrared fluorescence I given. We are coming to the most exciting part of the video, that is how the autobioluminescence occurs in a jellyfish. That is, I told you earlier, it was two proteins which is involving in this bioluminescence. It is a four-step process. Initially, the aquarin binds with calcium and it emits blue colored light, ultraviolet range to blue color light. It is collectively absorbed by the green fluorescent protein. Upon absorption, it goes for a modification, the change I told you. Later, it re-emits the green light. That is the fluorescence we are observing. The mechanism of action is diagrammatically given here. The aquarin which binds to the calcium, upon binding, it releases the blue colored light, which is absorbed by the green fluorescent protein, which is immediate to that green fluorescent protein inside the barrel. Then the structure goes for a modification, cyclization as well as oxidation. Then it becomes an effective chromophore and it releases or it fluoresces in a green color. By observing lower wavelength of light, it emits higher wavelength of light. The scientists have observed a very unique peaks which comes around nanometers. So there are two peaks around 395 as well as 475 nanometer. One is a major peak, that is 395 blue light. At 475 it is a minor peak and re-emits at 509 nanometer. That is a green light which we are speaking about. Hope so you might have understood the concept or detailed mechanism of action or how the light is emitted. So scientists extensively studied and found out various colors of fluorescence. So here some few variants are given. One is cyan fluorescent protein CFP, blue fluorescent protein BFP, yellow fluorescent protein YFP, violet enhanced green fluorescent protein sapphire, as well as cyan excitable enhanced green fluorescent protein EGFP. All they have found out. Throughout this video, we have studied a beautiful protein. Yes, it is extensively used in a biology and biotechnology field. So it is especially used in a imaging, that means to localize particular protein or organ. Especially we can localize the recombinant protein also. It is also used as a reporter gene or fusion tags in a recombinant technology. It is used in a pathogen interaction research and also research tool. It is used as a research tool. Fluorescence, resonance energy transfer, FRET analysis, even in fluorescent microscopy it is used, GFP is used, LFSM, laser scanning, confocal microscopy, it has been used. Wonderful identification, isn't it? The credit goes to the three scientists who have extensively worked in this, but they got Nobel Prize, not in physiology and medicine. Beautifully, they got the Nobel Prize in chemistry. Okay, so Osamu Shimomura, Martin Chalfie, Roger Weisstein, these are three people, they got Nobel Prize in 2008 for this beautiful identification. Osamu Shimomura is a key factor of identification of this. The finding is so unique, isn't it? So we have to be unique in every action, that is it tells about, so that we can glow in a future life. So even in career we glow. So be unique in every actions. So I hope so last 6.5 minutes you might have heard a good information. If so, like, share, subscribe my channel. Thank you for watching. Please read once more, watch again and again. Thank you.